Okay, good afternoon, everybody, and thanks for the kind introduction. Thanks for the invitation to be here and speak to you. I'm a computer scientist by training, and I'm at the Weizmann Institute of Science. My lab, for the past 8 years or so, has been focused on studies of the human microbiome and its relation to health and disease. So I'm very happy that Dr. Lustig just gave a segue to my talk on mentioned the microbiome. So the microbiome are the trillions of bacteria that live in our body. They're equal in our number of cells, but they actually have 150 times more genes. There's millions of different bacterial genes that we each carry compared to the 25,000 human genes that we carry. So by studying human genetics until now, we've been focusing on less than 1% of the genetic material that we carry within us. And the microbiome field is it's a very young field. Only in the past decade were we able to start studying it by advances in DNA sequencing that allowed us to probe it extensively. And as we started doing this, we and others have been discovering fascinating links, also causal links, as I'll demonstrate, between our gut bacteria composition and function and various health and disease statuses. And so over the past 8 years or so, my lab has been focusing on studying the microbiome, mostly in relation to various metabolic health and disease statuses, but more recently, work that I won't have time to mention today in relation to heart disease and also multiple sclerosis, cancer, and various other disease areas. The first work that we did and published several years ago was looking at artificial sweeteners. And the reason we looked at that is because of the known association between people who are overweight and diabetic and those who consume artificial sweeteners. But if you look at this correlation, then you don't know what the cause is. So you might think that perhaps somebody became overweight and then decided to reduce caloric intake and then go and consume these artificial sweeteners. But an alternative hypothesis is that perhaps these sweeteners themselves are in fact in part driving the obesity and diabetes phenomena. So we wanted to study this question. And I won't go through all the experiments that we did, but to make a long story short, what we demonstrated is that consumption of artificial sweeteners can alter both the composition and function of the gut bacteria in a detrimental way that can actually induce symptoms of diabetes. And perhaps the most demonstrative experiment of that is shown illustratively here. So what we did was to take gut bacteria from a normal mouse that never consumed artificial sweeteners. We grew the bacteria outside of the mice in the presence of artificial sweeteners and then transplanted them back into germ-free mice. These are mice that we grow in bubbles that don't have bacteria. And when we introduced these bacteria that grew in the presence of artificial sweeteners, we could induce symptoms of diabetes in the recipient mice. So the sweeteners were only seen by the bacteria grown outside the body. Outside the host in this case. And in preliminary experiments in human, we showed that if we take lean, healthy people that don't normally consume artificial sweeteners, we give them sweeteners in the amount allowed by the FDA for just a single week, most of them develop transient glucose intolerance. And when we take their altered gut bacteria and transplant them into germ-free mice, the recipient mice also, we induced symptoms of glucose intolerance and diabetes within them. We continued to do other work on supplements that I don't have time to go into. Recently, we published on what happens to when you take probiotics, whether probiotics colonize, and should you take probiotics after you take antibiotics. Well, we can talk about that perhaps in the Q&A. I don't have time to go in that in detail in the talk. Other areas that we examined were diurnal rhythmicity, circadian diurnal rhythmicity. And I believe we were the first to show that even though our bacteria live in the darkness of our gut, they also exhibit circadian diurnal rhythmicity. So about 15 or 20% of your gut bacteria go up and down also during the day. And that is coordinated both by feeding patterns and by our internal genetic clock. And we showed that these diurnal patterns are very important for regulating several gene expression, epigenetic, and metabolite patterns in us, in the human host, such that when you disrupt the circadian activity of the gut bacteria, you also can induce metabolic syndrome types of effects and affect even distal organs such as liver detoxification of drugs. Another project that I want to mention is a project in which we studied one specific phenomena in obesity and showed a causal effect for the microbiome in this phenomena. And this phenomena is the phenomena of why we wanted to study why is it difficult for us to maintain the weight after we lose it following a diet. And so many of you are familiar with this effect. It's also been termed the yo-yo effect of diet. So we decide to go on a diet. In the short term, many diets actually work. This was mentioned also before. And so we lose weight in the short term. But with time, we will gain back the weight. In fact, we will gain more weight. And so over time, over the years, we actually will be increasing our weight even though actually with every bout of this diet and rebound. And so we wanted to study this, and so to study this, we set up a mouse model in this case. And on one extreme, we had mice gaining weight on a high-fat diet, and on the other hand, we had mice gaining weight on a normal chow diet. And then we introduced another group of mice. These mice were given a high-fat diet initially, and then by various different means, they were forced to lose the weight until they had equal weight to the group that was on the normal chow, normal diet. And at that point, we gave them a second bout of the high-fat diet, causing them to increase weight again, and then to lose weight again. And we compared that to another group of mice that initially was not given a high-fat diet and was only given the high-fat diet exactly at the point in time when the first group lost the weight and now they had equal weight. And what we found is that on the same exact diet, the two groups of mice, one with a history of obesity and the other without a history of obesity, they had differential weight gain on exactly the same diet. And basically the group of mice that had a history of obesity gained more weight, significantly more weight than the group that saw the high-fat diet for the first time. And so until this point, this only recapitulates the yo-yo phenomena that we are familiar with in human. But now the advantage of having a mouse model is that now we can study this more mechanistically and gain insights into what's going on here. And to make a long story short, here too, we looked at many different parameters that we can measure in the mice host. And we look at physiological, immunological, metabolic parameters in these two groups of mice. And we could not find any distinguishing feature between them. The only place where we found a distinguishing feature between the groups of mice was in their microbiome composition. And the point that we looked at was obviously this point where the two groups of mice had equal weight, because this is the point where at this point we're going to give the two groups of mice exactly the same diet. One is going to gain more weight than the other. And so if you will, somehow the group of mice that had a previous history of obesity somehow is encoding that memory of previously being obese because it's now going to gain more weight on exactly the same diet as another group of mice. And what we found in the microbiome is that initially all groups of mice, as expected, start with very similar, very equal microbiome configurations. When we shift the group of mice to a high-fat diet, we see a shift in the microbiome composition. This has also been well documented before in the literature. But what is novel, and we found here in our studies, when we forced the group of mice now to lose weight, we saw a shift in the microbiome, but it didn't come back quite to the same point as the group of mice that did not have a history of obesity that were growing on a normal diet. So far this is, of course, only a correlation. We see a correlation between a different microbiome configuration in the group of mice that had a history of obesity versus the group that did not. But now, because this is mice, we can actually experiment and we can see whether we can establish a causal relationship here. And so we did that by several different means. First, we treated the mice with antibiotics as we forced them to lose weight after the high-fat diet. And that was enough to abolish the enhanced weight regain phenomena after dieting. You can see that now the two groups of mice, after antibiotic treatment and eradicating much of the gut bacteria, now are gaining the same weight. So this already proves that the gut bacteria has a causal effect on this weight regain phenomena. But now we wanted to ask whether we can also find means by which we can treat and maybe cure the mice of this enhanced weight regain phenomenon. So here too, we tried two different approaches. First, was to do a fecal microbiome transplant, so a transplant of gut bacteria from the group of mice that did not have a history of obesity into the group of mice that had a history of obesity. And that fecal microbiome transplant was also sufficient in order to abolish and remove the enhanced weight regain phenomena, and now the two groups of mice are gaining the same weight on the same diet. And so, if you will, this microbiome transplant is a cure in these mice, at least, for this phenomena. But a microbiome transplant is something it's very crude. We actually don't control exactly what we transfer. And so we wanted to see if we can establish a more targeted, more direct approach for that. And so what we did was to compare the metabolites in the serum, in the blood of the two groups of mice, and we found multiple metabolites that were different in the group of mice that were at the same weight, but one had a history of obesity and one did not. And specifically, we focused on two metabolites that are derivative of the flavonoid biosynthesis pathways. These two metabolites, apigenin and naringenin, were missing in the group of mice that had a history of obesity, so we supplemented the drinking water of these mice, and indeed, supplementing with only these two specific metabolites was also sufficient to now abolish and remove the enhanced weight regain phenomena, and was also a cure, a more targeted therapy for this enhanced weight regain phenomena after dieting. Okay, so now I want to turn into what I mainly want to focus on in my talk today, which is our long-term studies of the relationship between nutrition and our gut bacteria. And this project started 7 years ago. It's still ongoing. I'll share with you some exciting unpublished results from a long-term clinical trial that we're working on. And 7 years ago, the reason we started this was actually I never planned to work in this field, but out of personal interest, I started to read about nutrition. And as I was reading about this, actually, one of the books that I picked up was Gary Taubes's Good Calories, Bad Calories, and that was really an eye-opener for me to realize that a lot of what I've just been collecting from the media and what healthy nutrition should be is really false, and has really been motivated by a lot of different interests unrelated to science. And that really led me to read a lot more scientific articles in the field of nutrition and realize how, in my view, the science in the field was very poor. And so I decided to really shift my interest into a long-term quest for trying to understand what healthy nutrition is. And also, the more you look at it in the media, the more you see that we really have no clue on what healthy nutrition is, because it seems that we're changing that 1 week after the other to the general public. So if you look, for example, at the cover of Time magazine, 30 years ago, it said that cholesterol is bad for you. 15 years after that, the cover of Time magazine says that cholesterol is good for you. And you can see these changes and questions, very fundamental questions on what we should eat. Should we eat carbs? Should we eat fat? Should we eat dairy products? Is cholesterol in the diet good for you? Is salt good for you? We see articles, at least in the popular media, contradicting each other every other week. And I think that really also tells us that we don't have a good understanding of what healthy nutrition is. And so that's why we decided to study this. And as a computer scientist, I wanted to take an unbiased, data-driven approach to this problem. And really, the idea was to collect a lot of data and then study that data and see what we find and try to go at it with just a data-driven and as much unbiasedness as possible. And as we were thinking of what marker of healthy nutrition should we study and what we should collect data about, after a lot of thought, we decided to focus on blood glucose levels, and more specifically on the postprandial blood glucose response, or the post-meal blood glucose levels. So these are the changes in roughly the 2 hours after you eat a meal. This was discussed extensively before. So if you eat a meal that contains some amount of carbohydrates, your body would digest that, release them into simple sugars in the bloodstream. From there, your body would secrete insulin, whose job is to lower the blood sugar levels and signal cells to uptake the sugar and use it for energy. But also, as was discussed before, insulin would also signal cells to store the excess sugar and convert it into fat. And this is part of the mechanism by which we gain weight. In addition, if you have a huge spike in glucose levels and you have oversecretion of insulin, your glucose levels could go down to below baseline, causing you to feel more hungry, and by that inducing also eating behaviors that would cause you to eat more. So the relationship of blood glucose levels after meals and weight management, in my view, are quite well established. This is the carbohydrate insulin hypothesis model that was also extensively discussed here before. Now, I should mention that we decided to, I'll mention that in a moment. In addition to weight management, there's also relationships between blood glucose levels and specifically postprandial blood glucose levels and various disease areas. So obviously, they're important for the progression, development, and maintenance of diabetes, but they've also been linked to cardiovascular disease and to cancer, some in association, some with more suggestive causation relationships. So that should tell you, first of all, that blood glucose levels after meals are a very important clinical marker for weight management and disease. That's one reason why we decided to focus on it. But the second reason is that we can actually measure it quite easily and in a continuous fashion because of the availability of continuous glucose monitors that we can attach to people and use these sensors to measure blood glucose every 5 or every 15 minutes. So ideally, we'd prefer to measure insulin levels. We think that is, as was discussed before, that is a more causative marker of the phenomena that we want to study, but that is not possible in a continuous fashion to measure today. But in healthy people, the insulin secretion, by and large, follows glucose levels in the bloodstream. So glucose levels as a proxy also for measuring insulin, we thought is a good marker to study and one that we can actually study in a large collection of people. And so based on that premise, we initiated a project in Israel called the Personalized Nutrition Project. We recruited 1,000 participants, connected each of them to a continuous glucose monitor for 1 week, measuring glucose levels every 5 minutes. And during that week, participants logged on a mobile app that we developed all of their food intake. They selected from 10,000 different food items that we had full nutritional values for. And so at the end of this study, if you think about it, in 1 week, every person eats on the order of 40 to 50 small and large meals. And so from these 1,000 participants, we had about 50,000 blood glucose responses to meals. And this constituted the data that we were then going to study. In addition, we also collected a comprehensive profile from people that included various body measures, blood tests, medical background, and food frequency questionnaires. And what was most novel in this study was measurements of their gut bacteria by both 16S ribosomal RNA profiling and full shotgun metagenomic sequencing. To give you a glimpse of the data, here's blood glucose levels for one participant during the course of an entire week. These are measurements shown every 5 minutes at the top. The blue stripes, purple stripes represent when this participant recorded going to sleep. And if we zoom in, we can see glucose levels rising exactly after this participant reported eating either breakfast, lunch, or dinner. They rise to different levels after each meal, depending on what this participant had to eat. And we can take the area under the glucose curve in the 2 hours after this participant ate a meal as a single quantitative measure of the response of that participant to the meal. And as I mentioned, we have 50,000 such quantitative measurements from our 1,000 participants, and together with that, we have the full nutritional values of each meal that was consumed. So the first question that we wanted to ask is, what is the response of different people when they eat exactly the same food? And so this was a free-living study. We actually wanted to track the habitual diet of participants, but in order to compare and answer this question in the best way possible, in the breakfast, we actually provided all of the meals. These were very simple breakfasts, so either bread with butter, or fructose or glucose with water. The first 3 meals, the bread with butter, and glucose, were consumed in replicates by each person, and always in the morning after the night fast, so that we'll be able to compare. And this really allowed us to compare how the same individual responds to the same food on different days, versus how different individuals respond to exactly the same food. And so in this study, we provided all of these 7,000 breakfasts to our 1,000 participants. And what we found, the first very striking result shown on an unprecedented scale prior to our work, was that when the same person eats exactly the same meal on 2 different days, we see a highly reproducible response, not perfectly accurately the same, because you today are not exactly you tomorrow. It depends on also what you may depend on what you had to eat before, exercise, and so on, but very highly reproducible responses within participants for each of these standardized meals that we provided. But when we looked across participants, we saw dramatically different responses in different individuals' blood sugar response when they ate exactly the same meal. And that's shown by the histogram here. The x-axis is this quantitative measure of the postprandial glucose response, what we call a PPGR. The y-axis represents the fraction of individuals for each standardized meals that had a particular response. And you can see that these histograms are all over the place for every standardized meals. We have some people that had a very low response, others having a mild response, and yet others having a hugely high response. And if we look, I'll show you raw data anecdotally for 4 participants. Here's 4 participants eating the exact same bread that we provided, 4 slices of bread, 50 grams of available carbohydrates. 1 participant having essentially no response. If we didn't report eating the bread, we wouldn't see it by the continuous glucose monitor. Another participant having yet higher responses, another even higher responses, another one having dramatically high blood glucose levels after eating bread. And so this variability, it wasn't just that some participants responded more highly to anything that we gave them, but we also saw different types, different clusters of people with similar responses. So for example, in our standardized meals, we saw that over half of the participants, as you may expect, responded most highly to glucose compared to bread or bread with butter, but another 35% or so had a higher response to bread than they had to glucose, and yet another 15% had their highest response to bread with butter compared to the other 2. And although more rare, we did find instances where also different people had opposite responses, like these 2 individuals here, one having a higher response to glucose than to bread, and another having a higher response to bread than to glucose. So more often the case, we find that some people respond highly to something and others respond with a mild response, but on occasions we also see these complete opposite responses. So even before I continue, I believe that these results really have a lot of implications, so I want to pause on those for a moment and share the implications I think these results have. And the first one is what these results mean is that any universal diet that we'll ever give to the general public will always have limited efficacy, at least in terms of its ability to normalize and balance blood sugar levels. The one exception that I'll make is that, of course, if you go on a ketogenic or very, very low carbohydrate diet, so basically if you don't take in sugar at all, then of course you will not have any blood sugar or very little, if any, blood sugar increase, and then there won't be variability between people, and that may be a very good diet, but that diet, we know about this diet for decades, and that diet is very hard to follow. Those individuals who can follow it, it could be a very good one, but for the other 95%, as soon as you begin to eat even 15 grams, 20 grams of carbohydrates in a meal, you will have a blood glucose response, and as our data shows, that response will be different between different people. The second implication is that concepts that we've been using, like the glycemic index, are equally going to have limited efficacy, because take, for instance, bread, which has a glycemic index of 65. If you know how glycemic index is measured, it's measured by taking about 10 or 15 individuals, measuring their blood glucose response to about 50 grams of available carbohydrates from the product that you're measuring, and then averaging the blood glucose response of these 10 or 15 individuals, and that would be your glycemic index. So that's a very useful concept if all the individuals that you measured had very similar responses and you took their average. But it's a really not useful concept if the individuals that you measure are very variable, and although you can take the average and you can reach the same average by having very variable people, this average will be meaningless and in fact even harmful for those individuals that respond significantly more highly than the average. And predominantly what we see in our data is that for any food that we test, we see great variability across people. We don't see that different people have similar responses. And in other settings where we had the ability to measure multiple times the same meal of different people, we see here in the case of wholemeal bread, we see highly reproducible responses within individuals. These are different individuals on the x-axis, highly reproducible responses within people, but highly variable responses across people. And this is true for every single meal that we tested, for bananas here, for ice cream, omelet-based meals, granola, salmon-based meals, and schnitzel-based meals, which are popular in Israel. So really this demonstrates that there's a great deal of variability in blood glucose response of people. And of course, in our study, the next step was to try and identify the factors that associate with this variability, because if we can identify those factors, we thought maybe we'll be able to develop an approach that would predict personalized blood glucose response to people, and we'd be able to tailor diets to the individual that would balance blood glucose levels. And so here we turn back to all of the immense collection of data that we collected from each individual, and indeed we could find many different parameters in the blood that we measured and various body measures that correlated, that when we look across individuals, variability in these parameters correlated with the variability in blood glucose response to the standardized meals. But what was most novel in our study was many different associations that we found between the composition of different gut bacteria that people had, either measured by the 16S data or by the full metagenomic data, and also by the abundance and enrichment of different bacterial pathways, so different metabolic pathways that, not us, but that are carried out by our bacterial genes. If they were enriched or depleted in different individuals, those signals also correlated with individuals' variability in blood glucose response to these standardized meals. And so we wanted to see if we can take all of these many different significant statistical associations and correlations and somehow put them together into a more unified model that would be able to predict personalized blood glucose responses to meals. And so before I show you our approach, I'll show you what state-of-the-art was prior to our work, and that's to use just the amount of carbs in the meal to predict the blood glucose response to the meal. And so across the 50,000 different meals, each meal here is a dot, you can see the correlation between the amount of carbohydrates in the meal and the blood sugar response. You can actually see there is a significant correlation. You would not get this by chance, but a correlation of 0.38 means that you're explaining only about 15% of the variability in blood glucose response to food by the amount of sugar, by the amount of carbohydrates in the meal. So it's highly significant, but most of you would probably not want to make your food choices based on the quality of this predictor. But I should mention that actually insulin-dependent diabetics, this is what they have today available, and this is what they decide on how much insulin to dose after meals based on the quality of this predictor. And this is, in our view, why blood glucose management is very difficult in patients that depend on insulin. So we wanted to see whether we can take all these other parameters and improve upon these predictions. And so we used a machine learning-based approach. So these types of approaches are the same approaches that Google and Facebook use to put ads for you or to do web searches. We use it here in order to try and take personal data and predict blood glucose responses to meals. And roughly the way this works is that it's completely unbiased. It's a completely automated approach whereby we take all of the data that we collected. So for each meal, we have the information about its macronutrients, the amount of lipids, fat, protein, and carbs. And we have also information about micronutrients in the meal. To that, we also add personal parameters, so BMI, gender, age, blood tests, answers to medical background and food frequency questionnaires, and also parameters about the composition, which bacteria people have, which bacterial pathways they have. So all of that information can characterize a single meal. For that meal, we also have its measured response, blood glucose response, as measured by the continuous glucose monitor, and we accumulate all of these 50,000 instances that we measured. And what we do is we play this game where we take some individuals, we put their data aside, and we try to learn rules that would predict the response, the blood glucose levels that were measured based on these features. And so many of you probably are familiar with linear regression. So linear regression would be one type of approach that one can try. Specifically, that approach does not work here well. So we use a different approach based on multiple different decision trees. These happen to also be state-of-the-art in the field of machine learning. The details are not so important, but what's important is that we learn this model, we learn the rules on a subset of individuals, and then we go and we apply those rules to a different subset that was held out while developing the algorithm, and we evaluate performance. And when we evaluate that on a group of individuals, we see that we can increase the correlation from 0.38 to almost 0.7. So now we're explaining about 50% of the variability, and you can see on the x-axis the predictions by the model for each of the 50,000 different meals that we have. Each of the predictions is made by a model that did not see that specific meal while learning the rules and learning the parameters. On the y-axis, we can see the actual measurement. So it's not perfect, but it's dramatically better than using just the amount of carbohydrates in the meal. And we're now explaining about 50% of the variability across participants. In fact, it's quite similar if you recall how I showed you the same person responds to the same meal on different days. The correlation was about 0.75, 0.77. We're now achieving a correlation of 0.68. And we were very happy to see that after developing the algorithm, we profiled 100 new participants, and were able to also explain 50% of the variability in this independently held out, newly profiled cohort. So we're getting very significant improvements with the algorithm using all of this additional personal information, including the clinical parameters and the gut bacteria, and we're able to predict blood glucose responses much better. So the next step was to see whether we can take this algorithm and actually design diets for people that would balance their blood sugar levels. And so in the study that we published 3 years ago, we did a short-term intervention, and I want to share these results with you and then tell you about pre-planned interim analysis results that we have from an ongoing long-term clinical trial. And so in the short-term trial, what we did was to recruit 26 new participants, and we randomized them into one of two arms. In each arm, each participant received 2 diets designed by the algorithm. The diets by design had to be equal in the amount of calories, so breakfast, lunches, dinners, and snacks had to be all identical in calories. But in one diet, we asked the algorithm to prescribe foods that it thinks for that person would balance blood sugar levels, would result in a low post-prandial glucose response to meals. And in the other diet, for the same participant with the same amount of calories, design what we call a high PPGR diet, a diet that would spike blood sugar levels in that individual. And we then asked participants to follow each of these diets for 1 week, during which we connected them to a continuous glucose monitor, and we also profiled the microbiome daily. And the other group of participants did exactly the same, just the order of the 2 diets was different. And this was a double-blinded, randomized clinical trial, so neither the dietitians prescribing the diets or the participants knew which week they were getting which diet. So I want to show you the diets for one participant. So this is one of the participants, and these are 2 diets, one on the right, one on the left. Both of them designed by the algorithm. Both of them have exactly the same amount of calories in breakfast, lunches, dinners, and snacks. And you can take a look at these diets and maybe think for yourself which one was predicted by the algorithm to be the good diet for this participant and which to be the bad diet. And as you look at this, you'll notice that both of these actually contain some foods that would probably not be prescribed by your average dietitian in a week of a diet that is supposed to balance blood sugar levels. And I should mention that this participant and most participants in this short-term intervention were pre-diabetic. And maybe for fun, we could see by a show of hands who thinks the diet on the right is the diet that was prescribed as the good one for this participant. Okay, and who thinks the diet on the left is the good one? Okay, so almost half and half, maybe 60-40 for the diet on the right. Of course, this study was done in Israel, so it had to be the hummus and the pita on the good diet. And this was prescribed by the algorithm for this participant. And I'll now show you what happened when this participant followed each of these diets for 1 full week. I'll show you the full continuous glucose monitor levels. And this, in our view, is one of the most striking findings that came out of this study. So here's the blood glucose levels every 5 minutes measured for this participant on what we call the bad diet, this diet over here, the one with the sushi. And you can see blood glucose levels spiking to high levels after meals, in fact, reaching abnormally high levels after meals, indicating that this participant is indeed pre-diabetic. And now the same participant on exactly the same calories in every single meal, the diet with the ice cream, blood glucose levels for an entire week. We achieve full normalization across the entire week, in fact, achieving fully normal, healthy blood glucose levels after meals. So this is one participant. Here's another 2 diets for another participant. You can take a moment to see if you can guess. I gave it out before, right? We have the hummus here. So this is the good diet, predicted by the algorithm. How well did it work? This also worked very well. So dramatically lower blood glucose levels for this participant when following the good diet. And I should mention that in this trial, some foods were given to some participants on what was predicted to be the good diet by the algorithm, and the same food for another participant may have been prescribed on the bad diet week for that participant. And overall in this study, short-term intervention, we saw that for most participants, indeed, we saw a dramatic lowering of blood glucose levels after meals while following the diet predicted by the algorithm to be the good one. So what in terms of the microbiome? So I mentioned we followed this daily, and I will say that by and large, the microbiome composition of participants in this short-term intervention remained quite stable, so they were much more similar to themselves even after this short-term intervention than they were to anybody else. But nevertheless, although people remained stable, there were some small yet noticeable and consistent changes between people in the gut bacteria composition, which was interesting because each person was following a different diet. But what was common to the diets of what we call the good week is that, as I showed you, they by and large lowered blood glucose levels in participants. And this commonality was sufficient to induce several consistent changes in gut bacteria. And what was interesting about these changes is that not a lot is known about the underlying bacteria, but in cases where something was known, it seemed that going on a diet that balances your blood sugar levels induced beneficial changes to your gut bacteria. What I mean by that is, for example, this species here, this Rosberria species, where low levels have been previously in a study, not by us, have been associated with type 2 diabetes. These bacteria, on average, tended to increase in participants when following the good diet. And several of these associations with health of bacteria, the changes that we were able to detect seem to be beneficial. And the reason that's intriguing is because it suggests that perhaps following this diet is not only good for balancing your blood sugar levels while you're on the diet, but perhaps by inducing changes in your gut bacteria composition, perhaps beneficial changes, it may induce beneficial changes that would prolong and be there even after you go off of the diet. That's, of course, speculation, which we need to prove and demonstrate in long-term interventions. And long-term interventions is exactly what we decided to do after concluding this study, because, of course, we'd like to see what happens if we take a group of individuals and have them follow these diets for a longer period of time. Can we induce metabolic benefits in them that would be clinically meaningful? And so for the past 2 years or so, my lab has been engaged in a very extensive long-term clinical trial. This trial is on people with prediabetes. So we recruited 200 participants. The study is actually still ongoing. We're nearly 80% done with recruitment. We expect the study to complete by mid of 2019, but I'm happy to share with you some unpublished pre-planned interim analysis results. So we recruited, the plan is to recruit 200 participants and then randomize them into one of two arms. One arm will receive our algorithm-based diet, the one that I just showed you, where we profile participants and then prescribe for them diets that would balance their blood sugar levels. And the other arm would receive what's called the standard of care diet. This is the diet that follows the American Dietary Association guidelines, because these are the current guidelines, and this is the gold standard for comparison. And in this study in particular, because we recruited people with prediabetes and because it's well known that losing weight will also induce beneficial changes in glucose metabolism, we actually wanted to, as much as possible, maintain weight in the two different groups, so that we'll be able to evaluate specifically the differential effect of these two different diets on blood glucose metabolism. So what we did was, what we're doing is giving participants a caloric target which should balance and keep them at the same weight. Participants are still losing weight because every time you go on some diet you lose weight, but it's much less weight loss than you typically see in weight loss studies, and it's actually equal between the two groups. So what we are looking at as the primary outcome are glucose metabolism outputs, and we're looking both at changes in average glucose levels as measured either by the blood marker of hemoglobin A1C or by continuous glucose monitors, which I'll elaborate in a moment. And we're also looking at at these spikes in blood glucose levels, and we want to see how much we're able to reduce people's spikes in blood glucose levels below the critical level of 140 milligrams per deciliter. So the way this study is designed, so we have about 1 month of a profiling period where we ask participants to stay on their normal habitual diet. During that 1 month, we profile them, we measure their gut bacteria, we put them on a continuous glucose monitor, and then the intervention itself lasts for a period of 6 months. And during the 6 months, one thing that we're doing very uniquely in this study is we're connecting participants throughout the entire duration of half a year to a continuous glucose monitor. So it's about 20 different glucose sensors measuring blood glucose levels every 15 minutes throughout the entire duration of the 6 months of intervention. I don't believe this was data set like that is available previously. And we then have another 6 months of follow-up after the intervention is complete. And that's why what I meant, that when we have the full continuous glucose monitor data, what we can do is we can use it to just directly compute the average glucose levels. We don't have to rely on noisy measurements such as hemoglobin A1C. We, of course, are also measuring hemoglobin A1C, but we can also take the averages directly from the full time course data of the CGMs. We're also looking at secondary endpoints, so various metabolic endpoints like changes in weight, body composition, insulin levels, fatty liver, and so on. Some exploratory endpoints having to do with changes in the microbiome and blood serum metabolites. And what we also are doing quite uniquely in this study is we're having people log what they eat during the study. So in many diet studies, you advise people on what to eat, and you only take once in a while like these 24-hour recall questionnaires, and you really don't have a good idea of what people are actually eating. And so in this study, we encourage as much as possible participants on the mobile app that we provide to log as much of the meals that they can. And I wouldn't say that everybody fully logs, but if we look at the 70% of participants, the top loggers on both of the arms, both the standard of care and the algorithm arm, we have a very impressive amount of logging. So about 80% of their caloric target are being logged by the top 70% of loggers in the study. So we really have a very good idea of what participants are eating. In fact, over the course of the 6 months intervention, we have about 1,000 different meals that are logged by each participant, over 350,000 calories on average, and by the blood glucose measurements from the CGM, we have about 20,000 blood glucose measurements throughout the half a year of intervention. And it's a lot of data collected overall throughout the entire study. So what does it look like? So here's one participant. This is one participant on the algorithm diet. These are his blood glucose levels tracked every 15 minutes by the continuous glucose monitor in the 1 month prior to the intervention on the habitual diet. And you can see blood glucose levels rising here. Red indicates levels above the critical level of 140 milligrams per deciliter. And you can see these abnormally high levels after meals, indicating that this participant is indeed with prediabetes. Now, on the first month of intervention, this same participant, following the algorithm diet with the diet that he was prescribed, in this case, we were able to, and you can see here, this is glucose levels throughout an entire month. We're able to completely abolish all of these spikes in blood glucose levels. If we look at this participant throughout the entire 6 months of the intervention, which he completed, we see major reductions in the blood glucose spikes. If we quantify that, we see that prior to the intervention, this participant was spending about 2.5 hours every day above glucose levels of 140 milligrams per deciliter. On average, during the entire 6 months of intervention, this participant is now spending less than 5 minutes on average every day above this critical level. If we use the CGM data to estimate the changes in hemoglobin A1C, we haven't looked at the A1C data yet. That's more noisy. We're waiting for the full 200 participants, but the CGM data really gives us a robust reading of the average glucose levels, from which we can take formulas in the literature to convert CGM data into A1C, and we see an estimated projected reduction of about 0.4 of A1C in this pre-diabetic participant. And I'll say something about the meaning of such a reduction in a few minutes. So here's another participant. Again, the first month of profiling on the habitual diet. You can see many spikes in blood sugar levels. And during the 6 months of intervention, not entirely losing all of the spikes. Of course, there's also part of these may be some issues with the algorithm. More so, it's actually some issues with compliance that people for the duration of 6 months can't always eat only the meals recommended by the algorithm. But you can see if we quantify this, also this participant spending over 2 hours above the critical level of 140 milligrams per deciliter, and on our diet spending on average less than half an hour now every day, and also a projected reduction of about 0.2, 0.3 in levels of A1C from this intervention. Here's another participant, and you can also appreciate here the reductions in spikes in glucose levels. Here's another one. And if we look at all 47 participants who completed half a year of intervention, and we compare the blood glucose levels averaged, the top ones from the time period prior to the intervention to those during the intervention, for many participants, we see very large reductions in these blood glucose spikes after meals. In contrast, if we look at the standard of care diet, then we see typical participants prior to the intervention having similarly high spikes in blood glucose levels, as I showed you for algorithm diet participants, but now during the intervention, these spikes in blood glucose levels are by and large maintained. Here's another participant. And if we quantify this, then on average, we actually do see some reductions in the spikes in glucose levels in participants on the standard of care diet. In the first 3 months, it's a reduction of about 20% in the amount of time they spend, about 140. In the next 3 months, that goes away a little bit, and now the reduction is only about 5%. In contrast, if we compare this to the average reduction that we see across the 47 participants that completed the 6 months of intervention on the algorithm diet, we see a reduction of about 60% of the time they spend above 140, and this reduction is actually maintained also during the next 3 months of intervention. And the differences between these are highly statistically significant. If we look at changes in the average glucose levels and the projected in the estimated changes in hemoglobin A1C by these two groups, we see that on the standard of care diet, in the first 3 months, we see some estimated reduction, but then actually maintenance of hemoglobin A1C after a 6 months time period. In contrast, we see a projected reduction of about 0.25, 0.3 in A1C in participants in the algorithm diet. Now, to put these numbers of A1C in perspective for a population of people with prediabetes. Remember, we're not talking about people with diabetes. To put these in perspective, the best thing that we have to compare to is the very large study done by the NIH, published in 2002, what's called the Diabetes Prevention Program, the DPP study. 3,000 participants randomized into 3 different arms. One is a do-nothing placebo arm. Another is an arm provided with a drug, metformin, the first-line for diabetes drug. And then another, which is what they call the lifestyle arm. This is an arm where they provided 3 different changes, 3 different lifestyle changes. One is a dietary change, similar to the diet that we're giving here by the standard of care diet. But then they also did caloric restriction, and they also did enhancement of physical activity. And so what's very nice is actually our standard of care diet, which only had one of the 3 components of the lifestyle change diet of the DPP. It's actually somewhere in between. It's better than a placebo do nothing, but it's worse than the lifestyle because it only has the dietary component. Okay, so it is working. It's better than not doing anything, but it only has a mild effect. In contrast, the algorithm diet, only using the diet without caloric restriction, without enhancement of physical activity, is achieving significantly larger estimated reductions in hemoglobin A1C as compared to what we see on the DPP lifestyle arm. And if you think that these changes, even the ones that they saw in the DPP, are not significant, then in fact they could actually be hugely significant, because in the DPP study they actually followed people for 4 years after the intervention, and what they found is that this reduction or difference between the lifestyle arm and the placebo arm of 0.15 on average in hemoglobin A1C in people with prediabetes was sufficient to lower the conversion to diabetes, could lower the incidence of conversion by 58%. And so if in our study we're able to achieve an even larger effect just by using diet as compared to the DPP, then we're hopeful that with these diets in the longer term we can show an even lower reduction and conversion to diabetes. So I'll just very briefly mention that we also have a smaller scale study in people with type 2 diabetes. This includes both a short-term crossover comparison of participants to the standard of care and the algorithm diet, followed by a long-term intervention. And here too, in this smaller scale study, we're also seeing that on the algorithm diet, we're on the same participant, we're obtaining a much larger reduction in blood glucose levels after meals as compared to the standard of care diet. And now this is on people with type 2 diabetes. So with that, I'll just summarize the main take-home message from the second part of my talk. So I showed you the immense amount of data collected on 1,000 participants measuring blood glucose responses to about 50,000 different meals, from which our data unequivocally shows that different people have different blood sugar responses to identical meals. I showed you that we can quite accurately predict these personalized blood glucose responses to meals by incorporating personal parameters about people, including various clinical parameters about them and gut bacteria information. And I showed you both in short-term and also in long-term interventions that by applying these diets, we can balance blood sugar levels in healthy people, in people with prediabetes, and also in type 2 diabetics. Recently, these findings on personalization of blood glucose responses to foods have been replicated by a group at Stanford, and more recently by a group at the Mayo Clinic in a study that will be published in the next upcoming months. Overall, in our studies, we've tested and validated the algorithm on over 2,000 different subjects, and more recently, the science has actually been translated into clinical practice, and it's now available both in Israel and also here in the US. So with that, I will just put up the most important slide in my talk. This is the acknowledgement slide of the many people in my lab that really drove and contributed and led all of the work, extensive work that I told you about. I also want to mention my colleague and friend who collaborated with me on all these projects, Eran from the Weizmann Institute, and our various clinical collaborators. And with that, I'd like to thank you for your attention and happy to take any questions.
some sort of fasting protocol?
Do you want this?
Do you think incorporating an arm where you looked at some sort of individualized fasting protocol might be able to recapitulate some of these gains that you have? Because it would kind of take food out of the equation and you would just be looking at different responses to fasting. And because there's such variability in the responses to food, maybe we should be just encouraging fasting instead of, you know, the food. Do you understand what I'm saying?
Maybe in part. So I think, you know, fasting and intermittent fasting, I mean, those are, I think, very interesting and subjects of active research, and I think they can teach us a lot. And I think would be interesting to study, and we have some plans to study them as well. I think long-term, though, I think, again, like I mentioned with the ketogenic or very low-carb diets, I think those would probably not be sustainable for the vast majority of the population. And for those, we're trying to find solutions that would, I think, in our view at least, be more sustainable and therefore would have a broader impact on a larger amount of the population. Although I think for understanding mechanisms, all of these different diets, the ketogenic, the low carb, the fasting, are very fascinating to study, and we can learn a lot of insights and mechanistic understanding, and then see how we can apply them in broader terms.
Could you just comment on how difficult it is to identify all these parameters in an individual? I mean, is this a small cost in terms of finances and time, or is it a very difficult protocol?
For everything that we've measured?
Yeah.
Yeah, so I will say, so in the research, we've actually looked at very, very broadly, because initially we didn't know where the signal is going to be. So we looked at a facet of many different parameters. Now, after the science has been completed, our institute licensed the technology to a company, and for a company that is trying to give this as a service to a broad audience, to a large public, then the challenge was, of course, to see how to reduce this to something smaller. And now, by just questionnaires and sampling of the gut bacteria, which is on the order of just a small number of hundreds of dollars, you can get this entire profiling which is needed for running the algorithm.
I'm going back to the mouse, the yo-yo dieting mouse, and wondered when you were presenting that if you could hypothesize in a human, if a human went on a diet and got to their ideal weight, would it be an idea to do a round of antibiotics on them to destroy their microbiome at that point and see what happens?
Yeah. So that would be an idea. I would not recommend it because I think antibiotics also have many different other effects that we would not want to have. So they induce, they alter the composition of the bacteria. We've published work on the rate of recovery from that. You can induce resistance to antibiotics, which would have longer-term harmful effects. But I think I think the antibiotics was more a proof of concept that we did to demonstrate that the microbiome has a causal effect on this yo-yo phenomena. But once we identify, so we use it as a proof of concept. Once we identify that the causality lies in the microbiome, we can go and dive deeper and try to identify what components in the microbiome are driving this. And as I showed, in that case, we were able to even bring it down to 2 metabolites that, at least in the mouse model, were able to provide and then cure the mice of this phenomenon. So I think it's more to use as a proof of concept to then search for more targeted therapy with that approach.